Professor Simon Carding, who has a session for us called The Next Generation, which I guess is the young ones, and a panel discussion that he's going to lead. Professor Carding from the Institute of Food Research in Norwich.
Thank you. Right, so this is supposed to be a question and answer session, but I thought what we'd do is we start off by each of these young people here introducing themselves to you, telling them what they're doing currently with regards to their research and how that may relate to you, and then open it up for questions. And I'm going to try and let them do all the talking rather than me. So if we start with Daniel on the end here. Sorry, just push this button.
You don't press it.
I don't press it.
Don't touch.
Now I'm on. Can everybody hear me? Okay, well, good afternoon. My name is Daniel Vipond, and I am the first Invest in ME funded PhD student at Norwich, at the Institute of Food Research. And I think, Professor Ian Charles gave a very good introduction to Norwich and to the institutes and universities based up there. And I join at a very exciting time where there is a lot of change happening, good change, investment. And since I have lived in Norwich and did my undergraduate degree in Norwich, things have changed in that small amount of time. And particularly in the area that I am studying in, the gut microbiota. When I first started my undergraduate degree in biomedicine at UEA, the term microbiota I certainly had not heard of at that time, and the number of publications was pretty minimal. And just in between 2009 and even after I've graduated, the explosion in this area of science and how it relates to human disease has been quite astonishing, the range. So it makes it a very interesting area to get into and to explore. And so my project will be looking at those aspects as well as gut permeability. And so my project at the moment is looking at ways to develop an assay which will address that question: is there leaky gut, increased gut permeability in ME, and trying to validate that and using that in the clinic. But I'm not on my own. I also have a team around me of students who will introduce themselves shortly, who are from a medical background, who are also very much involved with various aspects of the project. So it's a real privilege to have a team of people around me. It feels a real teamwork, which is what I enjoy the most. So I'll now hand it over to Navina to introduce herself.
Hello, everyone. My name's Navina Navanidaraja, and I guess Bharat and I are the first Masters funded projects in Invest in ME are supporting. So by trade, I'm a medical student at the University of East Anglia, and have been very fortunate to be supported by Richard Peer and Invest in ME in my project, which is focusing on the gut microbiota in ME. And I share the same enthusiasm that Ian Charles conveyed this morning. I believe the gut microbiota has the potential to answer a lot of questions in the pathophysiology of ME/CFS. And what my project aims are to characterise just who resides in the guts of ME patients, and if this harbours any clinical significance as a future medical professional. The learning opportunities this year have been really immense through a collaboration with Cornell University and our research group. I was able to have a period of study at Cornell early this year for 3 months under Maureen Hansen's lab, where I learnt techniques that I'm definitely going to implement in my own project here. I believe that we can conduct a gold standard microbiome project from what I learnt at Maureen Hansen's lab, and I look forward to doing that.
Okay, Barrett?
Yep, so similar, I'm a master's student, so I've done my 4th year, got 1 more year to go, taking a year off to do this. This is actually my 3rd conference, so came about 2 years ago, and then talking to Richard Peer and Simon, they were talking about funding masters, and I was interested. So I began in September, finished in September, and the original plan was to help Daniel, who had already been doing sort of the ethics and starting the patient collection, was to get that done, get some samples and answer my research question, which I was given, which was assessing autoimmunity against neuronal antigens. So we did have ethical delays and looked like the sample collections wouldn't get done in time, but fortunately UCL and Fane were able to help us out and give us some samples. So about 3 weeks ago, I started in Oxford, and I'm probably going to be there till the end of August, and I'm working alongside Angela Vincent, who's somewhere in the crowd, and if you remember, last year she gave a very interesting talk. The lab she runs is the world leading in terms of finding antibodies against neuron antigens, so it's the place to sort of be to begin this mission. It's good in the sense as well they have a lot of experience there, so we can avoid making mistakes that perhaps have occurred in other diseases. Multiple sclerosis, they haven't found an antigen, and the various methods of looking may not have been the best. So in that sense, we can hopefully use the experience at that lab to make sure the research is directed in a way which can increase the likelihood of finding something.
Good. I should say all this is made possible by working with Amalak Bansal at St Helier, where most of our patient resources is based. So, Fane?
So, unlike my colleagues, I'm all by myself. No, that's a joke. Not for long. So, my name is Fane Mensa, and I'm also a PhD student, also founded by Invest in Me. I work in a group of Dr. Geraldine Cambridge at UCL, where we've been looking at B-cell phenotypes in ME/CFS patients. We've been seeing patients from 2 different clinics, one from Amalak Bansal, one from Dr. Saul Berkovich, who works at Royal London Hospital of Integrated Medicine. And to say that last year I came here for the first time without doing any research on ME, and now 1 year further, I think we've accomplished a lot on the B-cell phenotype, which will be shown by Dr. Cambridge later, and which I've also shown 2 days ago at the conference. And what I just wanted to say is that I'm really pleased that I can be part of such disease, which is unknown. And for young researchers, as we all are sitting here, I think it's really important to get everything explored, and I hope we will do, and I think we should do our best, and also encourage other students to explore the Invest in Me and explore the immediate field, which we will probably also be doing. And that's basically it. Thank you.
Great, thank you.
Okay. Over to you. Any questions you'd like to ask these young people? Stop there. There's one down here as well. Yeah.
We have an interest in this sort of work already. We've been doing something for the question is, if you've got an increased permeability, what are you doing to find out what's going through and escaping into the circulation and possibly into the brain as well, to exert some sequential effects, which are adverse effects, but usually. Are you going to pick that up? You say that the gut opens what goes into the rest of the body, which you don't want.
So that's what I'm trying to develop now, an assay that will address that question of gut permeability. I mean, there are already existing assays which look at, for example, serum endotoxin from gram-negative bacteria. Yeah. Of course, there could be other avenues for which that appears from other systemic infections, so it's not necessary. So it's a surrogate of intestinal permeability, which is obviously quoted a lot in the literature. Also, the lactulose mannitol test as well, which involves patients ingesting sugar test solution. Again, that has some limitations as well. So what I'm trying to do is look at systemic antibodies towards members of the gut commensal microbiota and using it as a basis for an improved method of addressing intestinal permeability, and also changes over a longer period of time, because, of course, the current methods provide only a snapshot in time, I think. And it could be that gut permeability is a dynamic sort of process, constantly changing. So addressing longer-term changes in the permeability status, I think, is more interesting.
Well, it's 10 to 1 again, isn't it, for you guys? Because that's the number of cells you've got to cope with in the gut. But that's very interesting. Thank you very much. I'll need to speak to you afterwards because we've got some work on a completely different angle in autism, which is also linked with disturbed gut microbiota.
Thank you.
How much, from the patient's point of view, do you think the problem actually lies in the extremely rigid and exclusive approach of conventional medicine? Because this approach of gut permeability in the biome has been the centre of complementary therapies for 30 years, very effectively. It's been discredited, poo-pooed along with the illness. How much of this do you think is the problem of the model of medicine with its exclusiveness and its rigidity and its inability to look at real connections, that this has taken so many years to become even regarded as a fruitful area of therapy when it's actually been effective for patients for so very many years in the alternative approach?
I'm not a gut person, so...
Come on, that's easy. That's easy, come on. Have a go. Go on, say what you think.
Where do I start? Does the answer lie in the next generation? Yeah. I mean, so I think the benefit of InvestME having such a sort of great group of scientists is that we've managed to have this sense of scientific merit instilled in us this year, and I think that's a great foot in the door. And why am I answering the question? I'm thinking. I also think that ME. So I don't actually know how to answer the question properly. Barrett, do you?
Well, I would like to say alternative medicine, complementary medicine, etc. There is a danger in that because it's not evidence-based and someone continues to use that, they may not seek medical help. I mean, of course, I understand in this patient population there is no medical help which you can seek at the moment, which is why people are going towards that route. In commenting on getting sort of complementary medications validated, that's a whole another issue and a whole another topic, so I don't know how much I can comment on that. But, I mean, my main point is the medicine in this country is evidence-based. If we're giving something to someone, it has to be proven that it can give a benefit to a wide range of people. Otherwise, we can't recommend it be given. So.
But we're also in this area of sort of individualised medicine, sort of tailoring it to the individual.
Yeah.
So, for instance, our angle, looking at the gut microbiota, we can tailor fecal microbiota transplants to these patients. An optimized therapy to the individual, that is a possibility. We can optimize therapies for these patients and tailor them to the individual with fecal microbiota transplants, for example, probiotics that.
I think that's the future, though. I mean, in the future, I could see perhaps we'll have our microbiota sequence as well as our own genome, so that at the moment it is rather a crude attempt. The fecal microbiota transplants, of course, have had huge benefit to patients with Clostridium difficile infection. But as I say, it's rather taking sewerage and, from a healthy donor, whatever that is, and then inserting it into the patient and then hopefully restoring whatever imbalance or dysbiosis existed, but just starting with characterising exactly what might be the difference, particularly in ME, what evidence there is for dysbiosis is where we're starting. But also not just looking at who's there in terms of the gut microbes, but what they are doing as well is another question. So it could be that we see no differences from our analysis of the communities that live there, but the next question is, okay, what are they doing? And I think that's the future.
Should I just pick up on that question you asked there? I think one of the issues is there is no... Sorry. Sorry. Yeah, my wife tells me I mumble as well. Yeah. No, I think part of the issue with the NHS is there is no standard test to diagnose leaky gut. I mean, the lactulose mannitol test is not used widely in the UK. I mean, it requires quite sophisticated analysis of a urine sample. So there is no test, and that's part of the problem. So it's anecdotal, it's surrogate, and I think you can misdiagnose it based on those criteria quite easily. So you end up treating the patient, with something they haven't got, or you're not treating them for what they do have. So there is an issue, and I think what we're trying to here is address that fundamental issue, or Daniel is anyway, with his assay. So we're hoping to have a much better indicator of what a leaky gut is and what actually is getting across the gut and into the systemic circulation in the brain. Yeah. So it's an important step, but it's, we'll get there eventually.
Have you used polyethylene glycols with mixtures at all?
Well, you can use those experimentally. I mean, they're used in animal models where you can detect transfer of passage of these large molecules across the gut. And you can, and some people have used them clinically, but it's not a diagnostic test, so nobody's using it widely. Yeah. Sorry? The question is, does anybody use polyethylene glycol to assess permeability? Yeah, it's more a science tool, really. A question down here? Microphone.
In a closely related area, I read the other day that there has been, in England, probably in the US as well, an explosion in the number of Crohn's disease and related IBS diseases. And I wonder if you, any of you up there have any idea why or how this may be coming about. And obviously there would be a tie-in to what seems to be an increase in ME around the world.
Right, so that could be due to better diagnostic criteria being applied. But I think there is an increase in certain pediatric cases of IBD, and that is being more accurately diagnosed. I mean, the Norfolk and Norwich Hospital has quite stringent criteria for diagnosing this, which hasn't been applied previously. So I think we're picking up a few cases that, well, quite a lot probably, that have not been correctly diagnosed. And that seems to be a common theme with lots of these GI-related diseases. What clinical criteria do you use to define you've got disease A, B, or C? It's very difficult. But yes, I think there is an increase in pediatric cases of IBD. I think that's pretty clear. Why? That's another question.
Well, I wonder if the panel has any thoughts in addition to the one you cite of diagnostic issues, if any of them have any thoughts.
Frank, do you want to?
The thing is, I'm performing research in a different field, so I'm performing research on B-cell phenotype. So I think the other three colleagues might.
Well, you're an immunologist. It's all.
Yeah,
it's an autoimmune disease, right? Yeah.
So if you have questions about B cells, I'm glad to answer them.
Yeah.
Have you any plans to look at people who've recovered from ME? Because in my experience, there are basically two types of patients. There are ones who get ME and who don't recover. And there are another group, and there's a substantial group of people who do actually have the good luck to recover. And it occurs to me that you should be looking at them also to find out what it is that has helped them recover.
To what extent do they recover?
Sorry?
To what extent do they recover?
Completely.
Okay.
And furthermore, this is obviously a very difficult proposition, but to look at people as a cohort and try and pick up the ones who then eventually recover to see what it is about the course of their illness which allows them to recover. I've got one or two patients in whom this has happened.
Yeah.
Yeah, I'd like to answer that question. It's a good question. I think what's really important in that aspect is that we look at, for example, disease duration or how long people might be ill or people recovering. And I think it's really important to have a cohort of patients in different stages of the disease. So with our markers, could be B cells or looking at other things, microbiota. I think it's really important to see what we can do in that case, because it's really important to stratify patients as well based on disease duration or other stratification methods. I think it would be really important to see how changes develop in years or over time.
That's one aspect we're incorporating in our study, to take a spectrum of disease severity with a special emphasis on those who are at the bedbound stage of the condition, and to really focus on actually getting some data from that group of patients.
Question for Daniel. You talked about characterizing the gut microbiota, Daniel. Presumably that means characterizing the biota for a Western population.
Yes.
I recently saw a media report about an American Indian population, I think South American Indian population, whose gut microbiota had been examined, and they were completely different to that of a Western civilization. I wondered whether that was something that was going to be potentially of interest to you.
Well, again, it's a completely different environment, different continent. It's known that our microbiota reflects the environment and the lifestyle we lead. So our diet in Western society, thanks to McDonald's and processed food, of course, feeding your microbiota this rubbish to a certain extent is going to have an effect, and that's characterized, documented in obesity and metabolic syndrome. So, even to the extent those who you live with, your own family members, you will share aspects of the microbiota as well. I mean, we all have a core microbiota, which is, I suppose, essential for digestion, foods, etc. But we all individually have a microbiota imprint. But as I say, yes, where you live, who you're with, and your culture, as was remarked at the colloquium in the last couple of days, or actually I think at Professor Carding's public lecture the other week, that those who eat with their fingers in those cultures share a high proportion of their gut microbiota. So the best control for our project would be to look at family members who are unaffected and to compare the patient's microbiota with their own family.
One of the concerns that was raised in the article that I mentioned was an extinction problem, whereby these South American Indians who then came into contact with Western civilizations were actually losing microbes in their gut, and those were potentially going to be lost forever, and they were potentially beneficial for Western civilizations to use in the future. And so we're potentially facing an extinction problem.
Mm-hmm. Yeah, sure.
Yeah, that's true. I mean, it's quite clear that of all the diseases that you'd like to name that are linked to the gut, the one commonality is the loss of richness and diversity of your microbiota. That's quite clear. So clearly losing species of bacteria in your gut is not a good thing. The question is, do you lose them, or are they there just laying dormant and waiting for the right stimulus to come along? We know from changing diets that you can get very rapid shifts in populations of your microbiota within 24 hours of going onto a different diet. But if you stop the diet and revert back, then they'll come back very quickly. So you may or may not be losing them completely. But if you do, that probably is a long-term health issue. Yeah.
So what's the potential for setting up a zoo for these things?
Well, there are people who do this. I mean, there's a research group at the Institute of Food Research very interested in this, looking at bifidobacteria, and these are very important in child health. These are conveyed to the infant through breast milk and feeding from the mother, and they're very important for initial infant health. And what her group is doing is look at the diversity of this bacteria in different animal species. So she's got people going to Banham Zoo collecting poo from pandas and giraffes and snakes and you name it, she's collecting it to look at origin, diversity and evolution of these bacteria. Because if we can trace it back, then obviously we can identify where the good bacteria are coming from. We can actually use that then maybe as a therapeutic tool. So we need to understand more about where these bacteria come from. And most of them probably come from the environment originally. The soil, the water.
Thank you very much.
Thank you.
One more, Sam.
Go on. Thank you. Very interesting topic, this with gut microbiota. For me, the big question is, which comes first, hen or the egg? Is it the dysbiosis which causes illness, or illness which causes the dysbiosis? What is your approach?
So this is the problem with any study looking at intestinal dysbiosis. Is it a cause or a consequence of disease? And you're completely right. Is the dysbiosis the primary trigger? Is the dysbiosis occurring parallel to the actual cause of the disease? Or is it a secondary kind of shift in microbial populations? And I don't think we're still at the point where we can distinguish that.
But do you have any opinion, kind of, when you start your studies? What is your hypothesis?
Right.
Which way it goes?
So I echo what Daniel said about what's a more sort of pertinent question to ask is what the bacteria are doing as opposed to who are there. And if we can find a way of sort of tying this along with the symptomatology of ME/CFS based on metabolic pathways, neural pathways, immune pathways, I think that may hold an answer for us.
There's a question at the back, if we can get a microphone up there. I mean, you haven't said anything about ethics and the problems of getting ethical questions resolved before you can do any experimentation with human beings. I mean, it goes on and on and on, and it's getting worse and worse. You know, it puts people off doing it now. But, you know, you've got to fill in about 500 forms. I mean, what's your experience?
My experience of that is that it's a growing industry, and it has significantly held up my own project, trying to get the necessary approvals, particularly within the NHS as well. Although we have Dr. Banzel on board and carries quite a lot of clout, we still have to get through the R&D process as well. And I wrote, and not emailed, because emails can go down the list and not purposely be ignored, but I wrote 3 times to the manager of that department to get the approval underway. And it actually took me driving down from Norwich just for 10 minutes to be at their door and say, what are you doing? And I did get it sorted out promptly afterwards, but it took just to going all that way for 10 minutes to literally say, look, I'm here, deal with me. And that's so frustrating.
Yes, question in the back.
I'd like to come back to your a little bit more qualified opinion or description of FMT than I'd hoped for. I was going to say I'm a layperson, but it's actually worse than that. I'm an economist. And an economist is known as the dismal science. And the joke about economists is that they lie awake at night wondering that things that work in practice can be made to work in theory. And that's very funny about economists, but it does strike me as quite similar to what I'm hearing right now about FMT.
So what is your question related to FMT?
My question is your qualified opinion on FMT really a function of you knowing it appears to be working, but not yet working out why it's working?
Well, it does work, but for a very specific clinical condition, which is recalcitrant C. difficile infection. I mean, it's amazingly successful. I think its cure rate is approaching 95%, which is far better than any drug that's available on the market.
But it's also working for ME/CFS patients.
Well, there are some isolated case studies coming out of Australia, but they are limited in terms of number, and some of these are not reported in scientific literature, so it's difficulty tracking them down. But it is potentially enormous in terms of therapeutic impact. I mean, it's been trialled by colleagues of ours at St Mark's Hospital for treatment of inflammatory bowel disease. And I know there are the groups elsewhere looking at potentially treatment of obesity. And we're interested in the Institute, the possibility of reversing aging. So, maybe not so crazy as you think. No. I mean, your gut ages before the rest of your body, which is interesting. So.
Well, there you are. There's some information before you go to lunch. Simon, we'll have to dash to lunch, people are indicating. Okay. Thank you very, very much to the panellists and to Simon.